go through the basics of smell physiology, and we're going to touch a bit on some anatomy. The objective of this class is for you to understand how smell is detected. So we're going to go through the transduction process as well as the central pathway, which parts of the brain it affects. The sensation of smell is known as a visceral sense, as it is very closely related to GIT functions. Physiologically, you note that taste and smell are always combined, because when you state flavor, you're stating a combination of taste and smell. Both their receptors are chemoreceptors. We learned that in taste, we see that stimulation by the molecules is in the solution saliva, while in smell is in the solution mucus. The stimulation comes from an external source, so these receptors are also known as exteroreceptors. So the sense of smell goes through the organ known as the olfactory epithelium. The olfactory epithelium is a combination of the epithelium itself plus the lamina propria. You see that the cells that transduce sensation of smell are known as olfactory receptor cells, and they are bipolar. And these cells are cushioned by supporting cells. So the receptor cells are the green ones, and then you have the blue ones there. Sorry for that sound. But you have the blue ones there that are known as basal cells. And then you also have the brown ones as supporting cells. Also, you have the Bowman's gland that is in charge of production of mucus. At the base or within the lamina propria, you have the olfactory nerve fibers going through there, and we'll talk more about it. But really, the place where the action takes place in transduction is within the nose itself, among the cilia of the receptor cells. So this diagram is elaborating, or more or less putting into perspective, where the olfactory epithelium is. So when you look at the nasal conchae, or conchae, nasal conchae, yeah, you will notice that on the upper portion of it, that's where we have our olfactory epithelium, and we'll talk about the details of it. And you see that the upper component of it is composed of an olfactory bulb, where within it is the olfactory tract. And right above this is the frontal lobe of the cerebrum. So getting it out and just blowing it up, we want to see right at the bottom a mucoid layer in blue, and then we have our olfactory cilia. Then we have the first layer known as the olfactory epithelium, composed of the dendritic end of the sensory receptors, and also the axonic end, and in the middle, of course, you have your nucleated component. Supporting it are the purple cells that in this diagram are also known as supporting cells. And then right at the base here, we have these orange-like structures known as basal cells. And then we have the now labeled as olfactory gland in charge of the production of the mucus. So what we see here is we see our axons piling up in groups, okay, to penetrate this bone. Now this is the cribriform plate of the ethmoid bone. All right, so they penetrate through that, and remember that between that and the olfactory epithelium, we have what is known as the lamina propria connective tissue, where you find the bulk of the olfactory gland. So after the penetration of these axons through the ethmoid bone, you see that they gather in what are known as glomeruli, and they come as so if cell number 1, 2, and 3 were together, they will synapse in one glomeruli, and we'll talk about more of that very soon. We also have cells that are known as mitral cells, and there are others known as tufted cells. We'll talk about them very soon. But at the end of the day, what happens is after they synapse on these mitral cells, the mitral cells are the ones then that form the axonal ends, form the olfactory tract. And this is what leaves the nasal area as the olfactory bulb. So what we now want to dwell on is just a few more features of this area, and then we'll go into the physiology. So one of the features that I like to emphasize is that you have an area of about 5 centimeters squared in the roof of the nasal cavity near the septum, being in charge of olfactory transduction, or that's where the cilia is. And the olfactory mucus is the place within this area where you have the nervous system closest to the external world, because these cells that pick out or that transduce the signals of smell into action potentials actually directly attached to the nervous system. So you don't have much of receptor organs that make up this area. It's the actual cells themselves that initiate these action potentials. The cells are about 10 to 20 million of them. So we have a lot of cells that go are in that place. Sorry for that. But what you should note is that each olfactory receptor is a neuron. That's the takeaway message. So each olfactory receptor is a neuron. Hence, this place is where you have this nerves as close as possible to the external world in the body. And there are quite a number. So as I said, most of this is repetitive, but it's important for you to realize that each of these neurons is a bipolar cell. It has this thick dendritic end with an expanded area that has got cilia, and the cilia projects to this mucus space, and each receptor neuron has about 10 to 20 cilia. Now the axons of the receptors pierce the plate, like we said earlier, and one interesting thing is that they have a half-life of about just a few weeks. We'll talk more about lifespans of these cells. So it's been observed that new olfactory sensory neurons are generated by the basal stem cells. And these odorant receptors, because of the noxious chemicals sometimes which are inhaled, may cause them to be replaced on a regular basis. So the nasal epithelium, including the olfactory cells, can be harmed. So to reduce the risk of olfactory neurons being harmed by these toxins, they are very limited in terms of lifespan, which is about 60 days. So because of this imposed cell death, the stem cells within the nasal epithelial layer differentiate into new olfactory cells. So in doing so, we have axons of the new neurons having to find their way to the targets in the olfactory bulb. So what do? They're able to do so by literally growing along the axons that are already in place in the cranial nerve. So this is the only place in the body where we continually make new neuronal cells. So that's a very nice anatomical feature to appreciate. A little bit more about the mucus producing glands. You see that the membrane is constantly covered by mucus, and the Bowman glands are the ones that are in charge of the production of this mucus, and they are also known as olfactory glands. They are just under the basal lamina of the membrane, and in this case we have turned it upside down to just show you where you find them right there. We won't go into details of the types of cells that they have, but this is just to appreciate that they are present, even though we have already mentioned. Feature I would like to emphasize on is now what we have been calling the olfactory bulb, where we find all these glomeruli, the mitral valves, and the axons of the receptor cells, as well as these mitral cells, forming the final nerve that goes to the brain. So axons of the receptors contact the primary dendrites of mitral cells and other cells that are known as tufted cells. Now they form complex glomerular synapses called olfactory glomeruli, which we will appreciate very soon in terms of physiology. But we also have some other cells that are known as periglomerular cells. These are sort of inhibitory neurons connecting one glomerulus to another, and the physiological impact of that is really just to ensure that you smell what you're smelling. So it will downplay other smells that you're really not sensing, so that one particular smell that is getting to you is sort of emphasized. So you have this lateral inhibition that goes on due to such neuronal arrangement. So we will appreciate this physiology in a few more slides to come. Talked about all that anatomy. I just want us to go through the basic neural circuits in the olfactory bulb itself. So I'll go through some names, go through how they are attached, and let's see if we can take it step by step. So the olfactory receptor cell itself, the one that is a bipolar one, has one type of odorant receptor project to one olfactory glomerulus. This is what we are calling the OG. So let's say you have this light purple or the purple smell. So you see that all the purple smells are going to link up in one olfactory glomerulus rather, and then you see that the pink smell is also meeting in another glomerulus. The CP here is the cribriform plate. Another feature that I would like you to appreciate is that the cells with one type of receptor project to different cells with another type, of receptor project to a different olfactory glomerulus. So again, it just pauses to what I'm saying. Pink must go to pink and purple must go to purple. You can't have pinks and purple mixing in terms of glomeruli. So if you're going to smell a jadot, it's going to have to go into one type of glomerulus, and if you're going to smell capenta, it will have to go into another type. So the sensations that are picking up capenta will meet up, or they are anatomically linked to meet in one type of glomerulus, and those that smell jadot, as you sense all of this through your life, they are also anatomically wired to meet up in another type of glomerulus. So another thing that you should note is we have what are known as there are several cells here that I want to introduce you to. One of them is the granule cells. So the granule cells are these ones that are yellow. Now, maybe let me take it up from the OG. What happens in the OG? So in the OG, or in the glomerulus, you have cells that are known as I think we just talked about the cells in the previous diagram, where we had tufted cells and mitral cells. So the mitral cells and the tufted cells are the ones that synapse in the OG, or in the olfactory glomerulus, and their axons then move up to the olfactory cortex. Then we have these granule cells that have no axons, and they make reciprocal synapses with the lateral dendrites of both the mitral and the tufted cells. So they're yellow here, and they're not a GR. So you see that they are on both types of smells. So they are synapsing on both the pink ones and the blue ones. And so what's the whole point of them synapsing on all those? So what happens is, at these synapses, the mitral, the tufted cells excite the granule cells by releasing glutamate. So you find that if you're smelling the blue one, it's going to excite these granule cells by releasing glutamate, and the granule cells will in turn inhibit the other side by releasing GABA. So the granule cells in turn inhibit the mitral or tufted cells by releasing GABA. So the blue ones are exciting the granule cells, and then you find that there's an inhibition that goes on. So whatever is coming from the pink side is sort of toned down, and the signal that is going to the brain is a blue signal, so to speak, or use the smell of blue. So the granule cells are very, very important. We'll talk about the periglomerular in just a minute. Okay, now let's look at our physiology step by step. So you have your odorants coming in with the air, and there are molecules within the air that are going to have odorant molecules that are going to bind to specific parts of the cilia. So the odorant receptors are located on the olfactory receptor, which occupy a very small part of the upper part of the nasal epithelium, and these are the ones that detect the inhaled odorant molecules. So there's initiation of action potentials there, and when the odorant molecules bind to the receptors, olfactory receptors are activated, and the action potential goes all the way to the glomerulus. Now the signal that are relayed in the glomeruli, well-defined microregions in the olfactory bulbs, will excite the mitral cells and forward the signal to higher regions of the brain with maintained specificity. The maintenance of specificity is due to the presence of periglomerulars as well as granule cells that we have talked about. But what you should know again is that receptor cells of the same type are randomly distributed in the nasal mucosa but converge on the same glomerulus, as I earlier explained. So what really is smell transduction? What happens? How does the action potential get initiated? Now, before I come to the diagram, I'll just go through my notes and state that the olfactory system has many different odorant receptors, and all the odorant receptors are coupled to G proteins, which are heterotrimeric. Now, the G protein is stimulated and triggers activation of adenylyl cyclase. Now, if we go to our diagram here, we can see adenylyl cyclase being activated, and the enzyme speeds up the conversion of ATP to CAMP. So when we have our CAMP there, this will cause the binding to action channels in the membrane cilia. So what do I mean by action channels? Here what we have calcium channels, and then we have some chloride ion channels that are down there, but let's see how each of them are affected. So this causes channels to open. Let's say when it binds to the calcium channels here, it will cause them to open, and you will have calcium coming into the cilia. So the influx of calcium is the one that's going to activate the chloride ions to open, and once chloride ions are opened, what is actually seen is chloride efflux, and there you are further depolarizing the membrane, or the inside of the cell is remaining even more positive. So then the action potential in this case will travel down the axon of the receptor cell, eventually meeting with other axons and forming the olfactory nerve known as cranial nerve number 1. So really what we're talking about, if I can just break it down, is you have odorant molecules that will bind to these G protein coupled receptors. Once they bind, it will cause the activation of adenylyl cyclase. Adenylyl cyclase will cause ATP to break down to CAMP, and CAMP will activate these calcium calcium channels, leading to influx of calcium, and the influx of calcium will then activate the chloride channels. Chloride channels will allow an efflux of chloride ions rather, leaving the inside even more positive, initiating an action potential which will then move up the bipolar cells. Something that we saw on the diagram earlier is the fact that the calcium channels are also sodium channels, so they can allow an influx of sodium. So this one is still the same mechanism. You have an odorant attaching to a receptor, which is G protein coupled, leading to the adenylyl cyclase activation, which will allow ATP to form CMP, and this will then activate the calcium channels. So the only thing that I wanted to highlight is the fact that they may also allow an influx of sodium. All right, now let's remind ourselves of what we have done so far. What we have done so far is we have appreciated how transduction of action potential occurs at the cilia level. We've also appreciated that there are millions of olfactory sensory neurons, and that each one of these has different odorant receptors. Now, each neuron projects to a specific glomeruli, which then synapses with mitral cells, and these mitral cells have periglomerula, which are sort of there to sharpen or focus olfactory signals. How? So as I said, if you have a pink smell coming in, you have the periglomeruli. I actually gave an example of the granular, but you also have the periglomeruli being activated. Once it's activated, it inhibits the other types of sensations or other types of odors. Thus, it will sharpen this pink odor going to the olfactory cortex. So it is all to sharpen and focus what you're smelling, so that once you smell something, it's definitely that, and it's not a is it that or is it this. On this diagram, I'm going to sort of give a broad idea of the central pathway, and then as we go on in the next few slides, we're going to break it down. But what I wanted us to appreciate from here is from the olfactory epithelium, you have these mitral cells and the tufted cells, and all of these go to specific components of the brain. Now, before I go on to all these brain areas, I want you to know that there's what is known as the vomeronasal organ. Okay, this is really emphasized in animals such as dogs, where there's heightened smell. These are just directly linked to mitral cells, and they go straight to mostly the amygdala. But for the other ones, let's go through the tufted cell first. The tufted cells, they take their action potentials to the anterior olfactory nucleus, as well as another one called the tubercle. The mitral cells are more widely spread. They take action potentials from the olfactory nucleus all the way to the tubercle, to an area known as the piriform, amygdala, and the entorhinal cortex. Now, the olfactory tubercle, the piriform, amygdala, and entorhinal all go through send potentials to the thalamus that then go to the orbitofrontal cortex. Now, I know that this seems so much, but we'll look at the brain and see how all this is spread out. And then from the same four areas, we also see that it takes action potentials to the front of the brain, or the frontal cortex. The amygdala area, which is involved in emotions, also takes impulses to the hypothalamus, while the entorhinal cortex is linked to the memory area, which is the hippocampus. So here we're looking at hippocampal actions, that's memory. We're looking at thinking, smell affecting your thinking. We're looking at hypothalamic sensations being affected. And so let's just have a look at the central pathway in another form. Okay, so let's break it down. What are we talking about? So what we are emphasizing is the fact that information is transmitted from the olfactory bulb by the mitral tufted neurons. Now, the mitral are the ones that send action potentials to a bigger level, or to a bigger part of the brain. So you have the anterior olfactory nucleus, as well as the tubercle, the piriform cortex, and parts of the amygdala and entorhinal cortex, whilst the tufted cells only send action potentials to the anterior olfactory nucleus, as well as the tubercle. We have a specific accessory olfactory bulb projection going only to the amygdala. Now, what you should note here is that conscious discrimination of odor is very much dependent on the neocortex, which is the orbitofrontal and the frontal cortices, while the emotional parts of olfaction is derived from this limbic projection. So we're talking about amygdala and hypothalamic influences. So here's another aspect of the central pathway. So we have the bulb that is located on the ventral surface of the frontal lobe. And then the axons project to structures within the limbic cortex, that is including the cortex in the temporal lobe that is associated with long-term memory. And then smell itself does not require a synaptic connection in the thalamus before connecting to the cerebral cortex. So you saw that there was so many attachments to the cerebral cortices before any of those could be sent to the thalamus by certain areas. So because of this, it can often be a potent trigger for memories, because of this intimate connection of the olfactory system with the cerebral cortex. It can also trigger some visceral reflexes through connections within the reticular formation. For example, some people would definitely vomit at the smell of another person's vomit. So we have triggers such as that just because of this very intimate innervation of the cortex. This diagram, because it sort of simplifies what we have been talking about. So from the orange component right on top, you have your olfactory bulb, and the blue is the olfactory tract. So first of all, you have a contralateral innervation to the hypothalamus that can go all the way down to the reticular formation for visceral responses to smell, such as the one that I'm just from talking about. You throw up because you've smelled somebody's puke. Sorry for that term. But we also have other innervations, such as those that go straight to what is known as the olfactory cortex. And this is found in the temporal lobe. And this is where you have the conscious perception of smell. From here, without going through this area, you see that directly you can influence the hippocampus, which is all about memory. So there's this direct stimulation of the hippocampus where you just smell something and you remember something else. But it can also come from perception of smell first. So you know that, oh my gosh, that's a rose. It reminds me of that. So from perception, then it will go to the hippocampal area. The last innervation that we see is this amygdala area where a certain smell will cause an emotional response. So certain smells may make some people angry. Mostly it's because it's tied to a memory. But certain smells will put somebody in a different mood. These can also influence the reticular ipsilaterally, the reticular formation for some responses, visceral responses to smell. So I think this diagram sort of puts into perspective the areas of the brain that smell has an influence on, while the first diagram sort of just gives us a broader perspective of how these fibers move. Before I finish this lecture, I want to just touch on a few more aspects. For example, we talked about the vomeronasal organ. So this organ is not well developed in humans, but it's there in rodents, and this organ is concerned with perception of odors that act as pheromones. Now, it is located along the nasal septum in a well-known developed organ. Now, this sensory neuron projects right to the accessory bulb, which we have talked about, and this is primarily linked to the amygdala and the hypothalamus. So you get a lot of emotional reflexes, so to speak, due to smells. So it's there in humans, but not very well developed. All right, now the receptors respond only to substances that are in contact with epithelium and that are dissolved in mucus. But we've seen that for some, you need very little to actually tell that that's what you are smelling. Remember the musk? Perfume? Yeah. Chloroform? You don't need much. Methyl mercaptan is one of the substances. Garlicky smell, you don't need much of it to actually know that that's what you're smelling. And just for your knowledge, you see that humans can recognize actually more than 10,000 different odors, and as you smell some more, you actually learn to differentiate them. Determination of intensity, however, is very poor. So for you to actually see a difference between perfume being there and too much perfume, it will take a lot of perfume for a person to put on. So remember, again, thresholds and discrimination, two different things. Okay, so what happens when you enter a room? The room is smelling of strong odor, and then you sit there for a few minutes, maybe many minutes, and it's not as bad. It's because of adaptation. So we talked about this during test, but it's also another reminder that even in smell, continuous exposure to even the most disgusting odor will decrease perception of that odor. So, and this is due to the fairly rapid adaptation or desensitization that occurs in the olfactory system. If you want the physiology, you can just remember that it is mediated by calcium activated via calmodulin on what is known as the cyclic nucleotide gated ion channels. So we see actually that when these channels, specifically the A4 subunits, are knocked out, adaptation becomes really slow. So it's been owed to these particular ion channels. So let's get a little bit more clinical. So what is the role of pain fibers in the nose, apart from telling you that you're in pain? So we have the trigeminal pain fibers as the ones that tell you or perceive pain through this area of your body. But they're also stimulated by irritating substances. So the reflexes of sneezing or lacrimation and several other reflex responses are initiated through these fibers. Okay, so abnormalities: inability to smell, anosmia; diminished smell, sensitivity, hyposmia; increased sensitivity, hyperosmia. So what could actually bring about some of these? So when it comes to anosmia or hyposmia, we're talking about simple nasal congestions or damage to the olfactory nerves themselves. Also be due to trauma, and this can cause fracture to the cribriform plate. Tumors as well, neuroblastomas or meningiomas can also cause this, or infections such as abscesses. We see that in Alzheimer's disease, they have this damage of olfactory nerves, so that also is noted in these patients. Aging is also associated with abnormalities in smell sensations. We have more than 75% of humans over the age of 80 who have an impaired ability to identify smells. What about enhanced olfactory sensitivity? So this is common. It's less common, but it's very common in pregnant women. They become oversensitive to smell. Then we also have distorted senses of smell, known as dysosmia. Yeah, so in dysosmia, we find that there are several odors, but you cannot identify which one is which. So it can be caused by several disorders, including sinus infections or partial damage to the olfactory nerves, and just poor dental hygiene. Now, the latest has been COVID. There's a very nice mechanistic exploratory article by Raphael. I think it's Raphael. Oh yeah, and I've put up an NCBI URL that you can just look for and see what he explains. He explains about 4 phenomena that could actually lead to the abnormalities that we see in COVID.